Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Shalom, Namo Buddhaya, Om Swastiastu, Salam Kebajikan. Good morning in Indonesia time, dear speakers and participants of the international webinar with the theme, Update on Veterinary Stem Cell, the State-of-the-Art Stem Cell Research for Clinical Application. My name is Hugolin Devi, and I will be the host for today. How are you? I hope everyone is doing good, staying safe, and staying healthy. It's so glad that we can meet virtually in today's international webinar. And today we are going to have several materials related to veterinary stem cell from our very competent speakers. And also, I'd like to greet our speakers for today, Assistant Professor Dr. Chenpop, Sawangmake, DVM, M.Sc, Ph.D, and Dr. Sirirat Nantavisai, DVM, Ph.D. Also, I'd like to greet our Honorable Vice Dean in International Affairs, Professor Mustafa Helmi, Effendi, DRH, PTA, PH. All right. Today, it's a very nice day for us to have our international webinar in here. And yeah, before we start our event today, let us take a moment to proudly sing our national anthem, Indonesia Raya, with the musical accompaniment. Please, for the operator to play us the musical accompaniment of our national anthem. Berdiri jadi pandu hidupku, Indonesia bangsaku yang berhati. Marilah kita bersatu, Indonesia bersatu, hiduplah All right, thank you everyone for your participation, for singing our proudly national anthem, Indonesia Raya. And yeah, as I have mentioned before, that today there is also joining with us the Vice Dean in International Affairs, Professor Mustafa Helmi Effendi. So I'd like to invite Prof. Helmi to give us the opening remark. Prof. Helmi, time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. The first of all, let us give thanks to Allah subhanahu wa ta'ala, who has given various pleasure on all of us. One of the flavor, now we feel it is blessing of health, so that we can hold this guest lecture properly. Furthermore, let me express my appreciation and gratitude And thanks to Dr. Epi M. Lukman, Master of Science, and also Professor Dr. Wijiati, Master of Science, and also the entire guest lecture committee who have prepared this much awaited event. This is very important for me to convey, considering that the Faculty of Veterinary Medicine, Universitas Airlangga, is working hard to achieve public recognition as a quality faculty in Airlangga University in implementing a quality management system toward the world-class university. The quality above are qualities that's balanced throughout. In Bahasa Indonesia, we call Tridharma Perguruan Tinggi, that consists of education, research, and community services, while maintaining the noble character within carry it out. The guest lecture theme, update on veterinary stem cell, the state-of-the-art stem cell research for clinical application. Of course, it will be useful for the development quality of postgraduate students in veterinary science in the future or in clinical research program. Finally, I would like to thank for all speakers for giving in the guest lecture held by Division of Veterinary Anatomy, with the hope that it will give enlightenment for us, especially those who are always involved in research, learning, and the application of the veterinary field in our respective life. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, Prof. Helmi, for giving us such a very welcome opening remark. And we are very happy to have you here. And also, there is Dr. Epi Muhammad Lukman, also join us with here, the Honorable Head of Divisions in the faculty. Welcome, sir. And all right, everyone. The next session is the most awaited session, the main session for today's event, which is the guest lecture. And this session will be moderated by Mr. Suryo Kuncoro Jakti, DRH MPet, Ph.D. So I'd like to invite Mr. Suryo to lead us the session. Mr. Suryo, time is yours.
Okay, thank you, Debbie. Good morning, everyone, and Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, I'm Suryo Kuncoro Jakti, and welcome to today's webinar entitled An Update on Veterinary Stem Cells, the state-of-the-art stem cell research for clinical applications. Before we get started, I'd like to remain a view item so you know how to participate in today's session. In this webinar, of course, you join the presentation by your own device, your phone, your tablet, or your computer. During the presentation, you will have the opportunity to submit text questions to today's presenters by typing your questions into the chat box. We will collect them and address them at the end of the presentation. Please don't forget, please indicate your question. Please indicate to whom your question is. In today's sessions, we have two distinguished speakers, our colleague from Faculty of Veterinary Science, Chulalongkorn University, Bangkok, Thailand. And for today's sessions, we have two speakers. The first speaker is Assistant Professor Dr. Chenpop Sawangmake, DVM, M.Sc, Ph.D. And the second one is Dr. Sirirat Nantavisai, DVM, Ph.D. Okay, without any further ado, let me start to the first lectures, but let me check first. Dr. Chenpop, are you already joined with us, or let me check first. Maybe he will a bit late. For the he with us. Good morning, Dr. Chenpop.
Good morning, Dr. Sayo. Thank you.
Okay. Thank you for joining us. But I will introduce you first to our participants. Okay.
Thank you.
Okay. Over that, I would like to introduce our first speaker. Our first speaker is Assistant Professor Dr. Chenpop Sawangmake, Doctor Veterinary Medicine, Master of Science, PhD. He is an assistant professor at the Department of Pharmacology, Faculty of Veterinary Science, Chulalongkorn University. And his research area are stem cell biology and translational therapy, bioengineering, molecular and cellular pharmacology, diabetes mellitus, bone tissue engineering, and also corneal regenerations. He will deliver the topic for today's sessions entitled Mesenchymal Stem Cells-Based Therapy, a New Paradigm in Veterinary Regenerative Medicines. Ladies and gentlemen, please welcome to Assistant Professor Dr. Chenpop Sawangmake, Doctor Veterinary Medicine, Magister of Science, PhD. For Dr. Chenpop, the time is yours. You have about 45 until 50-minute presentation times, please.
Thank you very much, Dr. Suryo. Thank you very much, our participants. May I share my screen? Okay, do you see my screen? Okay.
Yes, we got
again,
yeah.
Good morning, everybody. So sorry for my late. Yeah, I just understand that we start at 8:30. I'm sorry. Okay. As I got I think it's assignment from Dr. Suryo and his team for me to share some knowledge and some research that we are working on with you today. So the theme that we are talking on is about mesenchymal stem cell, but actually we work a lot on many types of stem cells. So I would like to share part of our work to you. The theme is today is about these things. Okay. Before starting, I think we need to understand something about our inspiration. Always when we talk with our team, we may recognize somebody to get us have a really good inspiration. So we always talk about one or two groups of laureates in stem cell biology that they got every big things in terms of their research. So the first group we want to mention is a group that they were the founder of stem cell research, I think. The group that got the prize in 2007, Professor Capecchi, Professor Sir Martin Evans, and Oliver Smithy Professor. So they are the first group that construct and establish the cell line. That time we are able to construct the embryonic stem cells, cell lines. This is a really big thing for us because by this innovation of fighting, we can have a powerful tools of the cells that can create a lot of platform for us to do in the in this time. And it's a very big thing for us to create the animal models like the chimeric models. Before these findings, it's quite difficult for us to create some models of disease. But after this finding, after the chimeric animals generation, so we are able to do this thing easily. Right now, we just call the company and then get it in our lab within a week. But in the past, it's quite difficult for us. And the next group is quite interesting for us because in 2012, two professors Sir John B. Gurdon and Professor Oral Sensei Shinya Yamanaka, they got the sharing prize on Nobel's prize. They found two things. For Professor B. Gurdon, he found that we can create the cloning platform. He's the one that do the cloning. He started with the frog, and he used the frog as a model, remove the nucleus out of the oocyte and put the nucleus new from some cells from the maybe tail of the tadpole, and then this becoming the new frog. It's the very beginning of the cloning generation. And for Professor Yamanaka, he said that with some reprogramming model, they are able to produce stem cells, which is the so-called induced pluripotent stem cell or iPS cells. So this is a breakthrough innovation, because in the previous, we don't think that we cannot get the stem cells from somatic cells or mature cells. But from the innovation from Sensei Yamanaka teams, we are able to do this with any cell type. Right now, when we search the papers, we will see a lot of publications say that we can use many of cell type and just reprogramming them to be the stem cell-like property. And this will be the platform for us, because right now we can harvest some cells from the patent, and then we can do some reprogramming and then attach the cells to the cells target that we want. Coming to our background, after this inspiration, we have to find our supporters. So we went to our supporters. I went to the School of Bioengineering at the University of Washington in Seattle with their taking care of Professor Cici Shashili and the team in Dentistry School, Corneal City. So we got a big supporter on this. And then we come back to create our unit, so-called the WISBIC, or the British Stem Cells and Bioengineering Innovation Centers. So we create this lab around almost 10 years ago. So I would like to invite everyone to join and, come see our website to see what we are working on. To just search the BSc in the Google, and then you can go into our website to share and see our activities. And also we are open our work for any collaboration. A little bit about our supporters. Right now we are having a lot of supporters from our faculty, starting from Professor Ipshok Anand and Professor Brungrod and Professor Kevli. They are the former executive board that support us a lot. And we have our strong relation with the dentistry school of Corneal City. This is a big thing, because in terms of human dentistry knowledge, they are very, very advanced in these terms. So we have a lot of help things from them. We have a strong collaboration with the Faculty of Medicine, Corneal City, Professor Sirirat. He's very good in system biology and antibody platform. Dr. Sukansar, very good in bioengineering, and team from engineering school. They are the team from the Department of Medical Bioengineering, Dr. Jutama and Dr. Pirapat. So by this team, we are able to create the platform of tissue engineering and bioengineering that can be applied for both human and in animal. And we have a strong collaboration with Professor Shashili from University of Washington, Seattle. The big help from Professor Kusar from Corneal City. He's a dentist working mainly on the regeneration of the oral disease. And we have a lot of knowledge from him and a lot of cells from him. And we have a new collaborator from Corneal City, Professor Santiago and Professor Nadine, on working with the oral issue. So as I told you before, that we create the research units, but anyway, to make our platform coming through the clinical service or clinical application is quite difficult for us, because we need a lot of supporters and a lot of budget. So we have to set the technology startup companies. We call this company as BioInc, because we imagine that we are the ink that can draw the dream for people to making the bio innovation come true. You can also check our details in the Google. So we use this platform together. We use the company plus the research center to drive the innovation, go to the clinical service as quickly as possible. Because if we wait too long, if we ask only for government funding, it will take more than 10 years. We cannot wait for that. We have to rush up. So come back to our unit. So I will show you something about the theme that we are working on. Actually, we are working on around almost 10 themes, but we show you around 3 main themes. So right now we are working mainly on diabetes treatment. So we try to set up the platform to, let's say, permanently treat the diabetes in animals. So we don't want to see the animals to get some insulin every day. It's quite not good for animals. So we try to do some platform that can give us the chance to treat diabetes permanently, or at least 1 year for one transplantation of the cells. Next thing is about bone tissue engineering. We want to set up the platform to treat bone defects by combining stem cell plus the tissue engineering platform to treat some bone defects. And the next thing is about the corneal defects. So we collaborate with ophthalmology school and the faculty of engineering to create the platform of the corneal sheet formation. Right now in our lab, we have 3 main principal investigators, me and Dr. Sirirat and Dr. Bartriwan, working on the platform of the disease like diabetes, bone issue, or corneal degeneration. We are going to go to see the work of the diabetes team at the first time. So this team led by our principal investigator, like I told you before, Dr. Bartriwan, and we have our alumni, and he is also our senior mentor, Dr. Suryo. And we have 2 postdocs working on this, Dr. Saranju and Dr. Park Poom. Right now we have 3 graduate students, Dr. Hong Thuan Tan from Vietnam, Dr. Eritar from Indonesia, Dr. Quinn from Vietnam, and Dr. Padera from Indonesia. We have 1 scientist support us on this. So let us inform you something about it, our thoughts on diabetes treatment, because we want to set up the platform to treat diabetes permanently. So we think about idea of transplant the beta cells that we can produce in the lab. So Dr. Suryo proposed this idea in the frontier in veterinary science. So we propose that we may use a platform to induce the cells from any stem cell type, M.Sc cells or the EH cells or iPS cells, and we drive them to the mature or the precursor of pancreatic lineage. And then we can use a platform to encapsulate them, store them in some vesicle or some device, and then put this into the parentheses. We want the platform that can keep our cells stable and not fragile, and then prevent the cell from leaking in or leaking out. And then this should be prevent us from immuno attack, and also can supply nutrients and let the secretion of the cells diffuse out from the beads and let some nutrients, oxygen and glucose diffuse into the bead. I think Dr. Suryo can tell you more in his session. I won't go deep too much for this. So from this thought, we first set up our platform to build a IPC from human dental pouch stem cells and human periodontal ligament stem cell right now, because we have a strong collaboration with dentistry school from University, so they provide us a lot of cells bank for us. We use the multi-step protocol to change the cells from somatic cells, drive them back to their very naive state, and drive them to the lineage of endocrine. And then around 10 days, we got the almost mature pancreatic beta cells. This is the first protocol for us. So at the time, we are able to show that the cells can secrete the glucose according to the, the cell can secrete the insulin according to the glucose challenge also. And they can, after we stain the cells, we can see the expression of insulin protein within the cells. And then we modify the protocol by the work from Dr. Bashir and we publish in BBRC again. By this mean we say that we can use like integrative protocol combining the genetic modification with the microenvironment manipulation to drive the cells toward the mature beta cells. So by this mean we can create a lot of colonies of the cells. We have to do this because we want to get a very high yield enough for us to get the cells enough for transplantation. And we're checking the genes. So we get a very good response of the cell differentiation. So we move to the work of the tutorial. So when we get the platform to create the cells, and then we move to the platform to encapsulate the cell. Dr. Sawangmake did some work about the encapsulation, and he's gonna tell you more in the details, but I will briefly explain this this time. Dr. Sawangmake said that by the encapsulation technique that he invented with the team, the encapsulation platform can preserve the cells in terms of their viability, their function, and it can get the glucose and the insulin that the cells create diffused in and out from the beads. And for the platform can help the cell survive for such a period of time. So right now we are on the process to testing the stability of the cell within the bead for a long time. Sorry, okay. And the latest work from our team that we got published in scientific reports, it's a work from Dr. Bashir and she can tell you more in detail in her session. In this work, we tell that we can produce IPC from canine M.Sc that we derive it from bone marrow and adipose tissue. So by multiple step protocols, we can produce the cells, and we can show that the cells can work properly and express the gene, a protein like insulin and glucagon from their colonies. And because Dr. Bashir gonna tell you more, because this is very big papers and very interesting, I think we have to wait for her for explain this. I will explain you a little bit on our ongoing work. I'm sorry, on our ongoing work. So right now we try to modify more on the Bashir protocol to properly induce the adipose stem cells from canine source to be the IPC. With the work of Dr. Quinn, we are working on driving the cells toward the IPC and get highest yield that we can do, and try to encapsulate them. From the standing, you can see that we can get the colony that have the expression of insulin and glucagon in the same colonies. This is good for us because we want to mimic the natural islets of Langerhans. We don't want to make a pure beta cell because sometimes the bodies need some balance between decreasing and increasing of glucose level in the body. And then the next work will be the work from Dr. Hong Chun Tran with the strong collaboration with Professor Hiroshi Egasa from Tokyo City. Professor Egasa provides us the iPS cells from mouse origin. So we use this as a platform to prove that this the model that we want to create a model that if we can harvest the cell from the patent and we drive it into the iPS in our lab and then drive to the IPC or beta cells and encapsulate them and transplant to the patents back. It will be the platform that we can use autologous cells to treat the patent. This is the way you want to reduce the problem of the cell rejection of the transplantation. So by this platform, Dr. Hong Chun Tran can show us that we can produce the insulin producing cells from the iPS cells. We can get a lot of colonies in the homogeneous size and morphology. And we plan to transplant this into the animal models to see about the efficiency of the protocols of the cell induction and efficiency of the transplantation platform. I think we can finish this work around early of the next year. And one more thing about the work from two colleagues from Indonesia, Dr. Ardeta and Dr. Nabila. So both of them just graduated from our master program. We got a grant from Red Cross Society to see the relation between the catalytic induction or catalytic occurrence in animal models that link to catalytic phenomenon in human with some diabetes or some defects of their body. So they have the concept idea that, so we know that when our body get in the stress situation for a long time, we have a low level of chronic inflammation. This is not good because it can induce a lot of bad things of inflammation, plus the stress in the gut line. So if we have the gut leak, a small leak of the cells, the junction of the cells, it can cause the leakage of some small antigenic molecule from like bacteria, like LPS or something, into the circulation and can trigger low level of inflammation throughout the body for a long time. So we create this model in an L10 knockout model and then induce gut leak in the animals. So we see that the big things happens. The first thing is it can harm the pancreas. So we have a reduced size of pancreas in the animal model, and then we have a small size and defects of the beta cells in these models. So by this means, we can conclude that with prolonged stress sedation in both human and in animals is not good for the health, because it can harm the beta cell function and beta cell morphology. So we try to revitalize the cells by doing the cocktails of the medium that can make the cell more healthier. So Dr. Ardeta can produce the cocktails that can preserve the cells and make the cell more healthy. And we are in the process of making the patterns of these cocktail formation. And the next thing that come from Dr. Nabila works. So we see that with this model, we get a defects of the bone formation and the bone morphology. In the catalytic model, we have a strong defects of bone. So it's quite interesting that when we have a strong stress, it can harm our bone formation. So we see the defect of bone marrow cells also. So these two works are on the process of submission for publication. Our next future is to produce a canine induced pluripotent stem cells, that we want to use this as a platform to produce the insulin-producing cells. So this work gonna be done by Dr. Padera and one of our graduate. Dr. Naklin is our undergraduate students that come working with us directly in the 4th year of his study. And we have two postdocs working on the making exosomes from canine M.Sc for treatment of some disease like the diabetes and the renal disease. Okay, this is our work of the DM team. So we're gonna go quickly to the work of the bone team. So the bone team led by Dr. Sirirat and Nantavisai, our staff, and we have 4 members of graduate students: Dr. India from Indonesia, Dr. Nabila, Dr. Steven from Indonesia, and one from Thailand, Dr. Tinutri. She is just graduated from the DVM degree, and we have one scientist supporting this. Dr. Sirirat proposed the themes of the M.Sc-based bone tissue engineering in some publications. So we propose that for in veterinary fields, we might have some chance to make the bone engineering of the bone from some M.Sc cells. Before that, people asked us that why we don't use like a pluripotent cell like iPS cell or ES cell, because they are very strong origin of the cells. We said that it's true that the cells from ES cell or iPS cell, they are very good in differentiation toward any cell lineage. But for us, in reality, it's quite difficult for us to maintain the ES cell colony or iPS cell colony for such a bone application. For us, we think that only MSC is strong enough to be the seeder from the tissue entering for the bone defects treatment. So we propose that 3 sources of the MSC from canine origin, like from bone marrow, from adipose cells, or from dental pulp or dental tissue origin. They are a very good candidate for combining with a tissue engineering platform to build the artificial bone. And then Dr. Rat did some work about some reagent like somatins that can enhance the cell proliferation and also the cells differentiation. Dr. Rat published her work in scientific reports, telling us about the behaviors of the M.Sc that we got from the bone marrow and the pulp cells during their osteogenic differentiation. So we use the high throughput platform like a bioinformatics to tell this. Dr. Sirirat gonna tell you about this in her session. So I just go roughly on this. So we show that with the cells that we derive from the cell bank, so we can drive them toward the bone origin, to the bone lineage. And with some proteomic platform of analysis, she can explain you more on this. We can conclude that these 2 cells, they employ different pathway to maintain their osteogenic differentiation. One employ a lot of system like the notch signaling, the BMP, the TGF, or Wnt signaling, but another employ some like a partly compelling with their MSC from bone marrow. So this explains the phenomenon that we have found long time ago when we try to drive many of cell origin toward the bone lineage. We found that they are going very differently. So at the time, we don't know why. And then we decided to collaborate with the team in faculty of medicine, Chulalongkorn City, to explore this. And then we found that, okay, they employ different things. So it's some kind of like a chip paradigm of our thinking that we have to tailor-made the protocol to that cells, maybe cell by cells, or maybe application by application. And the next thing is the ongoing work. So I will tell you a little bit about our ongoing work. We are planning to try to set up the platform to treat some oral disease like the periodontal disease. So that's the main idea, working on the effects of IO6 on the occurrence of the periodontitis and the osteotic differentiation potential of the PDL cells. Right now, we use the model from human cells. This is quite interesting for us because we know that in both human and in animals, the periodontitis is a big issue for the animals and human to lose the teeth. So if we can understand this and we can find some way to preserve the socket and preserve the teeth, it will be the new paradigm for animals treatment, because right now in severe case, we just have to extract the teeth out. It is a bad news for us because it affects a lot of their quality of life. So Dr. Minadia going to work on this, and she know a lot of things on the effect of IO6. Right now, he is working on the process of the lab work, and we hope that by this year we can finish this project, and then we can get publication into an journal. And Dr. Stephen working on some molecules that so-called OGP, osteogenic growth peptide. We use these for enhanced bone formation of the cells. We're using the cell from PDL tissue from human again, and we are able to show that with the optimum dose, we can enhance the bone formation from the cells according to treatment. And I'm sorry. Okay, and we can we plan to embed the OGP with some material, and we plan to see the cells on this material and then transplant the cells to the bone defects. He is on the work. Right now, he is the master degree student, and he gonna finish his work by this year, and we're gonna have him continuing study with us with the PhD program, so he can stay for us more than 3 years from now. And the next thing, I think it's the last theme for us for telling you today. We are working on the corneal formation. Dr. Sirirat and with Sky Help from the team of ophthalmology from our faculty. So we are working on making the corneal sheaths and corneal stromal scaffold. And we want to make them, combine them to the double layers corneal. We call it corneal equivalence because we try to mimic all appearance of the cornea of the animals, especially for dogs. And we try to transplant these things into the patterns that have the corneal defect. Dr. Sirirat finished the first demo, the first work of her project. And I think we're gonna have the next team coming and then takes the knowledge and modify the protocol to get the base material. And then we're gonna transplant this in animal model to see the effects of the transplantation material. Okay, this is all works of the bioengineering platform in our lab. Right now, we are working more on maybe food issue because we got approach from the industrial sector. The Thai food group, which is top 3 of the food producing manufacturer in Thailand, approached us to making a meat, the pork meat from the culture lab. So we got this offer, and then we decided to do this. So right now we are in the maybe second years of the project, and then we can produce the first prototype of a culture meat product from the pork cells. So we are working on this more, and then I think around 2 or 3 years from this, we're gonna see the first commercialized or the first batch of the culture meat product from the company. All right, I think this is overall of the talk today about our themes of work and the direction of our lab. I'm very happy for any question. Thank you very much, Dr. Suryo and your team. Thank you.
Okay, thank you, Ajahn Chenpop, for very nice presentations. I feel come back to our labs. 1 year has passed so fast. Okay, before we open the question and answer sessions, could you please stay with us until the second speakers? I know you are a busy guy, but please stay with us because we are still missing you.
Thank you. Okay, I will stay. Thank you. Thank you.
Okay, thank you, Ajahn. Okay, right now we're gonna move to the next speaker. Our next presenter who will deliver the topic is Dr. Sirirat Nantavisai, Dr. Veterinary Medicine, Ph.D. She's an instructor in academic affairs, faculty of veterinary science, Chulalongkorn University, and she used to be a head of veterinary surgery and dentistry unit at the small animal hospital, faculty of veterinary science, Chulalongkorn University. Dr. Sirirat research area are bone tissue engineering, stem cell biology and translation, system biology, and also bioinformatics. In this occasion, Dr. Sirirat will deliver the topic entitled Veterinary Bone Tissue Engineering, Proteomic Approach of Canine Mesenchymal Stem Cell Osteogenic Models. Let me remind all of you, if you have any question during the presentation, you can still submit the question through the chat box, and please indicate to whom your question is. Ladies and gentlemen, please welcome to Dr. Sirirat. For Dr. Sirirat, you have 45 until 50-minute presentation times. Hello, Pinong, are you with me?
Hello, okay, thank you very much. Could I share my screen now?
Yes, I got it. Not yet, sorry. Can you share your screen?
Yes, I am sharing.
Okay. Okay, we got it now.
Okay. Okay, good morning, everyone. Selamat pagi, right? Good morning, and please let me introduce myself again. My name is Sirirat Nantavisai, and recently I applied to be an instructor in academic affairs of Veterinary Science, University. Today, first of all, thank you very much to invitation to me, and I am honored to be a part of this event. Today, I would like to present an update of the veterinary bone tissue engineering in the proteomic approach of the canine mesenchymal stem cells osteogenic models. First of all, let me tell you about the background of our lab again. After I graduated from my bachelor degree, I was a practitioner for 5 years in dentistry and oral surgery unit at the small animal hospital, University. During this period, I found that maxillofacial bone defects cannot correct by general medicine and surgery treatment. After that, I am interested to do more research for the correcting bone defects. And luckily, I met the Professor Chenpop and our team in the Wisbig Center of Veterinary Stem Cells and Bioengineering Innovation Center. And we are done the research in bone tissue engineering, diabetes innovation, and the corneal stem cell and scaffold. So this is the lab is like my second home, and I love this place so much. Welcome everyone in our lab. Okay, and in this presentation, I will focus on bone tissue engineering and proteomic approach. Okay, since we know that the bone tissue is a very complex structure, it covers the outside of a layer with a periosteum, and it consists of the small column called osteon or Haversian system. In each column, it can connect with the Walkman canal and the Haversian canal. Moreover, in the side of the bone, there are have the 4 main cells. First is the osteocyte, is the mature bone cells that maintain the bone matrix. Second is the osteoblast, and is the immature bone cells that secrete the organic and component of the matrix. Third is the osteogenic cells or stem cells that division then produce the osteoblast. And the last one is the osteoclast, that is the multilineage cells that secrete acid enzyme to dissolve the bone matrix. And then when the bone defects occur, by the definition, the bone defects is the lack of the bone tissue at the body area. When the lack of the bone tissue is resulting in like a pseudoarthrosis, artificial joint that is not physiologically important. In that area, 2 parts of disease bone are joined with the fibrous tissue. Right now, they are divided in 4 types of the bone defects. The first is a limit defects. Bone fragment have the contact defects. Bone fragment have no contact, and the complete articular defects. The cause of the bone defect is mostly come from the trauma, surgery, and congenital musculoskeletal disorder. For many years, autogenous bone graft or free vascularized bone graft have been widely used for non-union treatment. and have long been the gold standard for repair and reconstruction of the bone. However, harvesting of the graft from the iliac crest is associated with the donor side morbidity and particularly associated with the chronic pain and infection. So consequently, it is still is a challenge for reconstructive surgery to reconstruct of the large bone defect. Recently, bone tissue engineering is an interdisciplinary field at the intersection of the engineering, biology, and medicine, and has emerged as one of the most promising approaches to develop the biological bone substitute that restore, maintain, and improve bone function. And then this shows the three main components of the bone tissue engineering: stem cells, scaffold, and biochemical factor. And right now, various stem cells have been considered as the potentially powerful implement for the field of bone tissue engineering, as they can generate a material which mimics the natural bone tissue. And this diagram shows that the cells, osteogenic cell resource can come from several sources. For example, from embryonic stem cells, from bone marrow, from umbilical cord, from fat, muscle, or tooth. And then when they combine with the culture system and biomaterial, they can construct of the pre-bone structure. And when they implant, they can generate the bone tissue. Since we know that embryonic stem cells and induced pluripotent stem cells are pluripotent cells that can differentiate into a broader range of the cells than stem cells that are isolated from the mature tissue. However, due to the difficulty for harvesting immunogenic, tumorigenic, and destruction of the developing life from the embryonic stem cells. So the other source of the cells, including adult stem cells from adult tissue called the multipotent stem cell, attract the researcher to investigate the potential to differentiate into osteogenic lineage due to easy to harvest, no harm done to the donor, non-tumorigenic, and non-immunogenic. So this picture shows the example of the osteogenic cell resource from the multipotent stem cells that can come from bone marrow, adipose, periosteum, or the skin. And our study focused on the bone marrow and dental tissue. And this is the table that we conclude in our review article. We have 3 tables that if you interest on this more, you can go to visit this paper. We have 3 tables that mention comparing between 3 cell resources from bone marrow, from dental pulp, and from adipose tissue. Compare between the sameness property, osteogenic gene marker expression, and advantage and disadvantage of canine mesenchymal stem cells. Let's go deeper a little bit of this. For the osteogenic gene marker, compare between bone marrow and dental pulp. For the bone marrow, it shows the upregulation from the osteogenic gene marker, and also from the canine dental pulp, then show the upregulation also. But this we still need from the data of the dental pulp because it's still lacking of some gene expression from the literature. For the advantage and disadvantage of canine mesenchymal stem cells. So from the canine bone marrow, the advantage is can self-renewable, multi-region differentiation potential, high yield of isolation. However, the disadvantage is the invasive harvesting and in vitro cell expansion difficulty. While the canine dental pulp show high proliferation rate, high osteogenic differentiation performance, but the disadvantage is low number of isolated cells and less evidence both in vitro and in vivo. This is a review literature from the cell morphology compared between canine dental pulp and canine bone marrow. Both of them are identified as a spindle shape cells that grow on the plastic in the monolayer. And this is the review article from the in vitro that we can find that from the canine bone marrow and canine dental pulp can differentiate into the bone cells by positive of the erythrocyte staining compared with the human dental pulp stem cells and human bone marrow stem cells. We also found some study try to make a big bone defect at the mandible and regenerate the bone tissue by using the autologous bone marrow plus with scaffold. In this study, they plus bone marrow stem cell with the beta-tricalcium phosphate, and they show the success of the bone regeneration repair of the canine bone defect by using the autologous bone marrow with scaffold. Interestingly, from this paper, they show the promising cell-based therapy from bone regeneration by using stem cell from the deciduous teeth and dental pulp also. After they create the bone defect and the mandible, they insert the cells with the scaffold, and the results show the positive of the bone regeneration after 8 weeks after transplantation. So right now we move to talk about the system biology. So far, the system biology and tissue engineering have been developed independently. But right now, this is show the paradigm that link between tissue engineering with the system biology. From the tissue engineering, we have the biomaterials, stem cells, and tissue microenvironment. And then what is the system biology function? The system biology is a global analysis with the several layers of the study. For example, they can provide a transcriptomic, proteomics, interaction network, or pathway database. And then they employ the computational science within the network algorithm and data analysis. And then when we got the data analysis from the computational science, it's gonna come back to drive the tissue engineering together. So right now, the tissue engineering and system biology is a very closely connect together and drive together like a circle. And this picture show the layer and the level of the system biology. Like if we need to study of the gene or the whole gene in the cell, we're gonna use genomics. For the mRNA, we're gonna use transcriptomic. For the protein, use proteomics. For the metabolomic to observe the enzyme reaction. And to all of them is to understand and can explain the phenomena in the cells or in the organ and tissue. Right now, several studies have examined the secrete protein produced by osteoblastic cells and employ the standard biochemical methods. However, these studies are limited in the number of proteins identified because of the non-global nature of this approach. So right now, the mass spectrometry-based proteomic is the large-scale study of the protein. It's after the biological function analysis, proteome profile, and comparative expression level of 2 or more protein samples. And moreover, it can location and identification of the post-translational modification and interaction between proteins. This is the workflow and overview of the proteomics analysis, validation, and functional analysis. Some review literature show the powerful of this proteomic by identifying the different expressed protein responsible during the osteoblast differentiation from human mesenchymal stem cells. In this study, it shows a lot of lists of the protein that involved in different functions during osteoblast differentiation, such as the protein folding, cytoskeleton, or the signal transduction. Moreover, the phosphoproteomics analysis of the human M.Sc during osteogenic differentiation show the powerful also. Since we know the phosphorylation plays a crucial role in the signaling transduction network, so the osteogenic differentiation explains the phosphoprotein that demonstrate the dynamic alteration in cytoskeleton. So the finding in this review literature is make sure the powerful system biology approach, and then it's gonna modify in our lab to study more in this. Since we have cells, we have system biology, and then we know that during the cell differentiate into the lineage, into the bone lineage, it have the several osteogenic signaling involved during this process, such as notch signaling, Wnt signaling, TGF-beta signaling. So to apply to the clinical application, we interest to study more and deeper with the signaling to control the cells. And we hope that if we know about this signaling, we can control the cells and use in the real clinical application in the future. So this is the overall of concept of this study. Since we know the bone is a complex structure and we have the pain point of the bone defects, and we know, okay, the gold standard to treat with the bone defects is autogenous bone graft. However, it still have the limitation of this protocol. So we have the bone tissue engineering to overcome of this problem and employ the global study regarding the differentiation potential and behavior between two cells, and employ the system biology to know the function of the protein and the protein interaction, and can explain more about how to control the cells. And we hope that if we know the big data analysis of this, we can convert to use our fundamental knowledge to correct of this pain point. So this slide show the canine bone marrow and canine dental pulp collection and isolation. This is the different way to get of these stem cells. From the bone marrow, we can collect from the bone marrow and isolation with the density centrifugation. And when we get the layer of the cells, we culture and expand in the media. In contrast, from the dental pulp, when we have the tooth extract in the clinic, we add in the tooth transport and then move it into the laboratory under a sterile technique. We explant the pulp tissue and culture to expand the cells. To compare between the osteogenic lineage, we employ three important of this method. The first is osteogenic mRNA expression, runs to osteoric, osteocalcin, osteopontin, and collagen type 1. Moreover, we observe in the enzymatic activity level, we do the alkali phosphatase activity to prove of the osteoblast function. And the last one, we do the ECM calcification by using the one causal staining in the late state of the mineralization. And to globally analyze compare between two cells, let me explain more in this step because this is very important and I think this is useful for us to understand this. I divide in the step in the three parts. The first is cell culture and sample preparation. The second is fractionation and SCM-SNS. And the last step is the big one that I got in the big box, is the data analysis and interpretation. For the cell culture and sample preparation, it have many steps inside. For the fraction and LCM-SNS, we can employ the fractionation kit right now and do the MCLC and SMS. At this time, I employ the Orbitab to get the result and big data analysis with the Proteome Discoverer 2.2 and do with the David to analyze our protein function. Let's go deeper a little bit in each box. For the cell culture and sample preparation, the important step, the first one is osteogenic induction in vitro first, and then lysis of the cells, do the reduction and alkylation, digestion, peptide quantification, and labeling with the dimethyl. This is a chemical label to know where the protein comes from. So for the two lysis of the cells, we use the SDS in the lysis buffer called TEAB and add the protease inhibitor and make it homogeneous with the sonicator, and then break the disulfide bond with the T-cep and protect the disulfide bond rebound together. This step called the alkylation and digestion the protein into the peptide with the trypsin. And when we get the peptide inside, we quantify with the quantification kit. And then we label with the dimethyl. This is a highlight. We label 3 samples of this experiment with the dimethyl labeling. We have control, this means undifferentiated cells. We have day 7. Day 7 is mean day 7 after induction. And day 14 is mean day 14 after osteogenic induction. And each type of the peptide we label in different dimethyl molecules, including the control we label with the light molecule is plus 28 Dalton. Day 7 we label with the medium molecule plus 32 Dalton, and that day 14 we plus with 36 Dalton. So this is very powerful because we can know that where is the peptide come from, and we can make a quantification of proteomic result. After do the labeling, we have the fractionation method. In this study, we use the high pH fractionation. It have one word is called orthogonality. Orthogonality is the proteomic profiling that we can have the deeper to analyze and get more protein identification. However, it takes time and it have if you imagine that if you don't do the high pH fractionation, you're gonna have 1 tube of the sample and you do the 1 tube, just 1 tube. But if you do the high pH fractionation, you're gonna have maybe around 8 tubes, and per 1 tube, you're gonna use 90 minutes for LCM SMS. So it takes time. So you have to concern about this also, that it is okay for your research or not. But for me, I want to identify deeper and deeper. So I decided to use a high pH fractionation to fractionate of our peptide to be 8 fraction, and then go to let the sample to the LCM SMS. This machine, we use the Orbitab that have the electrostatic field. So the peptide can go along with electrostatic and calculate with the mass per charge ratio. After we got the data, we do the big part of the data analysis and interpretation with the program called the Proteome Discoverer 2.2. And after that, we analyze the function of the protein in the David program. Next, after we have the result from the global study from the proteomics, we select some potential signaling to prove of the functional of this signaling. So we found that Wnt signaling, Notch signaling, TGF signaling, and BMP signaling is the most promising from our result. So we do the inhibitor to block in each signaling. For example, when we use DKK1, Notch we use DAP, TGF we use SP431542, and BMP inhibitor for knockin and dorsomorphin. All of them is the canonical inhibitor and is the very interesting result they see together. Okay, I have around 8 figure results. The first one is proof that canine bone marrow and canine dental pulp show the mesenchymal stem cells property by show the morphology at the figure A and D is comparing control bone marrow and dental pulp. It show the fibroblast-like cells. Moreover, it show the mRNA expression relating the stiffness, LEX1 OX4 and proliferative gene Ki67. Positive for the CD73 and negative of CD45 colony formation, and they can show the potential to be multilinear differentiation of lipogenic, chondrogenic, and osteogenic from 2 cells. So it can conclude that canine bone marrow and canine dental pulp in this study is show the M.Sc property. And then when we got the cells, characterize the cells already, we observed the canine bone marrow and canine dental pulp about the osteogenic differentiation. At day 7 and day 14, we collect the result of the L3 activity, gene expression, and mineralization. For the ALP activity, osteogenic induction in both 2 cells can upregulate significantly compared between control. And for the gene expression, in the osteogenic group of the canine dental pulp and bone marrow, both of them show the higher significant differentiation during osteogenic induction. Moreover, in the mineralization part, canine dental pulp and canine bone marrow show the positive of the von Cossa staining. And interestingly, at the day 14, the last state of the induction, canine dental pulp show a lot of mineralization area compared with canine bone marrow. So it can be concluded that the canine bone marrow and dental pulp possess a different osteogenic differentiation, and canine dental pulp looks superior of osteogenic differentiation than canine bone marrow. And then this slide is used the volcano plot that reflect the distribution of expressed protein by osteogenic bone marrow, is one, and canine dental pulp. So we can see that the down-regulate this side, up-regulate this side. We can see that in the bone marrow, we can show the up-regulation of the protein pattern, both of the 7 and the 14. However, in the canine dental pulp, we can see the tend to down-regulate instead. So it means that it shows the different protein expression pattern between canine bone marrow and dental pulp. So we do explain more about the heat map with the dendrogram. We cluster the cells into 5 clusters. This is a canine bone marrow, this is a canine dental pulp, day 7 and day 14. We can see that, for example, in the cluster 5, it shows the red and red color in the both time period. In the red color, it means the upregulation of the protein expression in canine bone marrow, while in the canine dental pulp at the cluster 5, it shows downregulate and downregulate both time points. So it means that it's a distinct protein expression pattern between 2 cells. And moreover, when we do the Venn diagram, the 4 Venn diagram, it is we select the protein, we add the protein in the program. And if the protein is the same, it's gonna show in the intersect area in each time point. So we can see that all of them, all of bone marrow, at day 7 and day 14, osteogenic canine dental pulp at day 14 and day 7, only 10 protein that is the same. And around 160, this is unique from osteogenic canine bone marrow at day 7. Around 86 is unique from the osteogenic canine dental pulp. So it means that in these 2 cells and in different time period, it's totally different protein protein reaction or protein function. And this is go deeper with a heat map that illustrate the providing comparison of the significant protein. So we have the signaling cascade, we have the development signaling pathway, we have the miscellaneous signaling and extracellular matrix organization. You can observe it more in our publication, but I will explain a little bit of this table for you easier to follow us. We have osteogenic canine bone marrow at day 7 and day 14. And we have osteogenic canine dental pulp at day 7 and day 14. And we list the protein name here. And this is the number of the level of the protein expression. So if it down regulate, it's gonna appear in blue color. And if it this is up regulate, it is appear in the red color. So we can see that a lot of signaling involved during the osteogenic differentiation in both two cells, but different protein. And interestingly, a lot of extracellular matrix organization strongly involved in during osteogenic differentiation in canine dental pulp that we can see from the strong red here. Moreover, we have the identification with the cell cycle, DNA replication, metabolism of RNA, another metabolism, something like oxidation, carbohydrate, lipids, something like that, and organelle organization and cell-cells communication here. So if you're interested more, you can observe one by one of the protein that involved during differentiation. That is very interesting, and this is very powerful of the data information. And this figure, when we have the gallery, like the data information already, we do deeper about the signaling. So we make a pie chart here to observe about the signaling that involved during the osteogenic differentiation at day 7 and day 14, compare between canine bone marrow and canine dental pulp. And we conclude in this pie chart. After that, we select the important signaling, and we do the functional analysis by using the inhibitor. So we divide the experiment into 3 sets. The control is mean undifferentiated cells. The osteogenic is mean the cells with osteogenic media. And the osteogenic plus with functional validation is mean osteogenic media plus with signaling inhibitor, and observe at the 7 and the 14. And this is a result. Canine bone marrow and canine dental pulp. This is day 7, this is day 14 of the canine bone marrow. This is day 7 and day 14 of the canine bone marrow. For example, when we know that notch signaling is, some review literature mentioned about it is important to induce the bone. But in the osteogenic condition of the canine bone marrow, when we block the notch, but it contrasts, but it can enhance the bone mineralization here. And for the BMP, we can see that it's totally blocked the mineralization here. This is a very interesting. And for the canine dental pulp, after block with the DK1, DAP, or SB, or knock-in, it can enhance the bone mineralization, while for the dorsal morphine, it can block the canine dental pulp mineralization. This is the last picture that I do in my publication. Let me zoom a little bit of this. Okay, this is so when we know that the which signaling that involved during the osteogenic differentiation between two cells. So we do the tree diagram. This is a comprehensive understanding on the signaling dynamic. Tree diagram is the analysis of the tree function validate signaling that tracking the protein relation at the tree time point during osteogenic differentiation. So we have this one, this graph is day 7 compared with the day 0. This is day 14 compared with the day 0, and this is the day 7 comparing day 14. The red one is come from upregulation, and the yellow one is mean the unchanged protein, and the blue one is mean downregulated protein. In the green box of this is come from the protein of the, let me zoom in a little bit. The protein of the canine bone marrow, and the pink box is from the canine dental pulp protein. So we compare between Wnt signaling pathway, not signaling pathway, and BMP related signaling pathway. And after that, we conclude the signaling in each cell type that Chenpop mentioned before into figure. And this study already published in the scientific report. The topic is system biology analysis of osteogenic differentiation behavior by canine mesenchymal stem cells derived from bone marrow and dental pulp. And thanks for Ajahn Tailak and Ajahn Thanapum, Ajahn Prasit, and Ajahn Sirirat to help me to have this paper. This is very nice for collaboration with them. Okay, for the conclusion, this is an in vitro model of the osteogenic differentiation. This is a high throughput quantitative proteomics and validation assay of the candidate osteogenic signaling to obtain the comprehensive understanding of the protein behavior upon the osteogenic differentiation of the canine bone marrow and canine dental pulp. And this is the most comprehensive proteomics-based analysis of the osteogenic canine dental pulp to date. And the result of the present study indicate the numerous different behavior between canine bone marrow and canine dental pulp toward osteogenic lineage. These findings reveal the confirmation of the regulating osteogenic signaling pathways to support the mass spectrometry analysis. And this is a useful understanding for the canine M.Sc osteogenic part and suggest the trend of the NCSC based on tissue engineering. So again, with our lab, we do a lot of work, and we have the hard work, and also we have the party work. The party, also a lot of party. And welcome again for everyone to visit us at the www.cuwisbig.com. Okay, thank you for your attention. Terima kasih.
Okay, thank you, Dr. Sirirat, for your nice presentation. It's a very tough work for your during your dissertation time, right? Okay, from you, we learned two things. The first ones, by system biology, we can explore the potential signaling. And after we found the potential signaling, we have to validate that candidate of the signaling itself. Maybe after that, we can discuss more. Okay, we are now going to take a question and answer sessions. As a reminder, for all participants, you can still submit your question in the chat box, or you can just raise your hand and you can talk directly to the speakers. Okay, before we get started, I will invite again Dr. Chenpop. Are you still with me, right? Okay.
I'm here.
Okay, let me check in the chat box first. We have some question from our colleague. From the first questions, I will read it for you. How is stem cell development in Thailand? The first questions, and the second one about the implementation of stem cell, especially for clinical application in veterinary medicines. Maybe Ajahn Chenpop or Ajahn Sirirat, you can answer. That's
I think I will answer it first and then we'll ask Sirirat to add on some details.
Sure.
Getting to the question, how is stem cell development in Thailand about the application in clinical practice? I would say that right now we are in the process of study in the research center. According to the FDA law in the country, it's very, very strict. So every product needs to be registered. Only one that we can do with our registration is the tailor-made treatment, like we harvest the cell from the patent and modify something and put it back directly to the patent. This can be so-called tailor-made application, which is allowed by law, by the FDA regulation. But if we want to make a commercialized product, like we put the cells in the vial, waiting to be used for some patents and distribute to some facilities, this might be considered as a biological products that need regulation and need registered by the FDA law, and it's very strict process. When they need like a GMP facilities, they need the ISO facility to test the product. But anyway, we are working on this to make it come true. This is the answer. Thank you.
Thank you, Ajahn. Maybe Ajahn Sirirat will add more.
Yeah, that this is I mentioned about I am lucky to meet Ajahn Chenpop. That is the same passion together. So we want to do the research and then make it to be the clinical application. So I think it is important to find the team that have the same passion too.
Okay,
Yeah.
thank you.
Sirirat, can I add on something?
Yes, sure.
This about the direction of the research, because according to our career path, we are at the Bursa University, so we need to make the paper. But if we want to make clinical research and clinical service, it's quite difficult to get the publication. So we need to very, very organized on this.
Okay, maybe it will be related with the next question from Dr. AP. Dr. AP asking to Ajahn Chenpop, how did you build a team, recruitment process, how long does it take, and something like that, and get financial support, also build a network with a company? He asked about that.
Yeah, I think those can help me on answer this, but I would put some details. We start almost 10 years ago, and at the time, we have to have a direction very clear at the first beginning, because it will affect a lot of our direction. At the time, we don't want to make the papers. We just want to make the clinical application come true. So we decided to build a team by recruit most of the practitioners. So we want the DVM persons that have the heart of scientists. We don't want the DVM with the heart only like practitioners, because they think differently from innovation makers. So we need to combine the one that have a strong skill in clinical practice with the one with strong skill with the research, and combine them to be the one persons. But by this means, they can accept anything that challenge for them. They don't wait for the guideline from the society. They don't wait for the product from the company, but they make it by themselves. This is the first thing that we need to form. And then the team, if you have strong organization, strong outcome, it's very attractive for people. They're gonna come to meet you, and you have a big team later. And for the grant, I think it's challenge for us, because when we want to do the big thing, you need a lot of money, right? So companies, governments always ask how many publications you will get for them. In Thailand, they have the rate, like if we have 500,000 baht, we need to deliver them at least Q1 or Tier 1 publications. So this is quite challenging for us. So we decided to combine government grant with industrial sector grant together to build papers for our career path, and to build the innovation that can apply for the practice. I think it's not too difficult, but you need to plan carefully and then find a good stakeholders. and good colleagues to make the achievement come true. This is my idea. Maybe the Sirirat can add on some information. Thank you.
For me, because I start from my like a position in the small animal hospital. It is a practitioner, general practitioner in the small animal hospital first, and then I found that to build a team, it should be come from the the strong connection with another collaboration too. So for this lab work, we try to make a connection and collaboration with the international standard to move us to be like the global standard. So it is important to link with another university and another section or another department to help you to go to the goal of your lab. And for the add more the information about the company. When the first time that we do the research, we just add a little or a small lab work, but we do very hard work, and we try to make our outstanding. So when we have the work show to the publication, so the company will know you're powerful, and they're gonna go to contact you, something like that. So you have to think in the future too, that if the company come with you and talk with this topic, you can accept or not. So you have to have your direction.
Okay, thank you for Ajahn Chenpop and Ajahn Sirirat. The key point that we can learn is the collaboration. So we have to find the collaborators. And I have a comment in terms of government funding. I think the condition is quite similar with our country in Indonesia. If we got some government funding from some funding from the government, for sure they will ask the publication, how much publication that we can produce from our research. And maybe Ajahn, because most of the participants here also professor in the university, could you share how to manage the students, because you have many experience about how to recruit the students, how to handle the students, especially graduate students. Can you share to me? Maybe it will be beneficial to us how to build a laboratory and how to build a teamwork in lab as well.
It's difficult question, Sayor, actually. Actually, I'm very young in field of research. Let's say that, because compared to senior professor here, you in university you have a lot of senior professor. I think they have a strong experience more than me. But in my opinion, I just think that we have to we cannot do everything by ourselves. We have to build persons, build a team, set the system, and let the system work. The thing that we said and have done is that we build a strong leaders or a champions like Dr. Sirirat, Dr. Rachuwan, Dr. Suryo, or newcomers from our lab and new collaborators. Then we put there, give them a chance and the power to make some decision by themselves. So let them grow, let's say like that, let them grow, and then we just observe. And we have to talk very much on the direction of the thoughts, because sometimes people want to do the thing that they want, but if it's not go along with the lab direction, we need to talk and let them decide which way you want to go, because if we have too many directions, it's maybe the weak points of the lab instead of their strong point. So build person, build a system, let the system work, and then let them grow. I think this is the way. How about that, Ajahn Sirirat?
How about you, Ajahn Sirirat?
Yeah, I totally agree with you. Because when the Dr. Suryo asks this question, my idea pop up. Set a team. This is the keyword. To manage the many students in the lab.
Okay, thank you. We have another questions from our colleague, Dr. Eka. The question is for Dr. Sirirat, related with the bone, I think. Is it possible for the old animal with a bone defect that located nearby the joint cavity use therapy with a stem cell from mesenchymal, from bone marrow mesenchymal stem cell? If possible, how about the synovial fluid of joint cavity during recovery of the defect? Do you get the point?
Let me think of this. I think. So the question is about if the bone defects located near the joint cavity.
Yes,
and use the therapy with the stem cells from bone marrow.
Bone marrow, yeah.
So this is the first question, yeah, right? So if the bone defects occur, so the stem cells from bone marrow show the possibility to correct the bone defect. Anyway, from the old patient or the old dog is gonna have maybe 3 sources of bone marrow. The first is come from the autogenous, it come from themselves. But anyway, from the old patient, it's gonna affect the cell proliferation, I think. So the second choice is use the allogenic. It's from different patient. And anyway, it can come from the stem cell bank. But we have to concern about the interaction of the immunology. And the last part is really from the xenogenic, it's different species. Something, for example, from if we have to treat with the dog, we got the bone from the pig, something like that. So we have to concern and consider about this if we're gonna use the bone marrow stem cells. It should be come from autogenous or allogenic or xenogenic, something like that. But it's possible to treat with the M.Sc to the bone defect. And the second, I think the second answer, question is, if possible, how about the synovial fluid of joint cavity during recovery of the defect? I'm sorry, I don't get totally understand on this point, but in my understand, after we correct the bone defects and the joint and the synovial joint fluid can recover or not?
Yes.
This is the question.
Yes, I think that's one.
Yeah, I think it can be recover, but it's gonna take time because it's gonna old patient. Ajahn Chenpop, do you have any?
Actually, I think so. I think so.
Okay. How about Dr. Eka? You got the answer already, or you can ask more if you want. We have plenty
because time. The old animal, I think it's difficult to produce the synovial fluid of synovial. Yes, thank you for your answer.
Okay, we still have a lot of questions. The next question is from a student in Airlangga University. It came from Winda. I have a question, especially for Dr. Sirirat. In the slide, it has been written that the source of the stem cell can be taken from the bone marrow, adipose, etc. And also, each protein has different results in treating bone defect. So, do its case has its best stem cell option to be used, or how to choose a specific source of stem cell so we can treat a certain case and get the best result? Thank you. I think relating the screening of the donor about this one. Okay, Pinong, you can answer.
Okay. From bone marrow, adipose, and also each protein has different results in treating bone defects. So, do each case has its best stem cells option to be used? Let me think. also.
Okay, maybe you can just share to us regarding your study, because in your study you explore you use if I'm not mistaken, you use dental pulp, bone marrow, and another type of stem cell, right? And you use a system biology, and you find a lot of pathway, a lot of potential pathway, a lot of potential signalings. Maybe you can just share which one the best in terms of the bone defect. If you have in clinical application, if you have a patient with a bone defect, which one the source of stem cell will be the best?
Oh, okay. Because I am interested in the canine dental pulp stem cells, and I want to compare between another cell source also, that I select the canine bone marrow because this is the standard cells and a lot of information background of this. And then when we compare between two cells, in each topic, in each subtopic, such as osteogenic differentiation, proliferation, M.Sc property, it seemed like a canine dental pulp show superior potential than canine bone marrow. So we do suggest to explain to do the research more of this, but right now if I get cheer up of the canine dental pulp. But anyway, the canine dental pulp, due to the accessibility of the patient, it means that if we want to use the dental pulp from this patient, it means it's difficult to extract the tooth from this patient also. Comparing with bone marrow, we can draw the bone marrow from that patient, right? So this is the gap from the bone marrow and the dental pulp when you want to use in the clinical application. Anyway, several studies support that when we use the allogenic canine dental pulp stem cells, it has several immunology support the evidence that in the first week of transplantation of canine dental pulp, it showed the pro-inflammatory cytokine increase, but it's not show the clinical sign to rejection. So this is a further analyze or further study in the animal model for the canine dental pulp transplantation.
Yes.
May I add on something? Yeah.
Okay.
Actually, it's quite difficult to choose which one is the best cells for use, right, Dr. Sirirat? We have many factors to consider. The banking of the cells, the availability, accessibility of the cells, whether it's difficult to get it or not, and how difficult we can grow them and make them in a bank. So according to experience, we know that each cells, they have some different behaviors. For bone marrow, it's quite easy compared to covering to each other cells. It's easy to access. We have a lot of cells when we do one harvesting, but to maintain them, to grow them is a bit difficult. Comparing to the pulp cells, even we get a small amount, but they can grow very quickly. Or adipose cells. At first, people think that the adipose cell is not interesting, but right now we found that they are some kind of good cell source for us to do more. So I think the best answer might be, it depends on application you want to use and about the cell number you want to get from each cell resource. Yeah.
Okay. How about Winda? Do you still have any question? If you still have any question, you can just ask directly to our presenters. Okay, or you can type if you want. Okay, we have another question from Dr. Purnama. The question is for Dr. Chenpop. Regarding your recent study in corneal regenerations, which one is better compared to the golden standard to treat corneal dysfunction using endothelial keratoplasty?
I think right now we haven't tried comparing study. And this issue, I think that's another Sirirat might answer you better than me. We are on the process of making the corneal equivalents to mimic the real corneal, but it's for sure it's not good as some natural materials. Actually, we have collaboration with some companies. They build the sheet from some collagen fiber, but technically it's still difficult to arrange the cornea from collagen fiber to make it clear enough. Right now, we might not focus on the clarity of the surface, but we might focus on the healing rate and the scar formation instead. And the clarity might be the next thing that we want to working on. Yeah, it's still difficult to say right now.
Yes. But if you are still interested about the corneal defect, we still have Dr. Sirirat, and she will present her work tomorrow morning. So just stay with us tomorrow morning. Okay, and another question. We still have another question from Dr. Kusdiantoro. Maybe he is our colleague from IPB University, if I'm not mistaken. To Dr. Sirirat, interesting result. What is principal difference of induction signaling between bone marrow M.Sc and dental pulp M.Sc that make dental pulp M.Sc superior than bone marrow M.Sc during osteogenic development and also regeneration? Thank you.
Okay, thank you for the question. The principal difference in induction signaling between two cells is BMP signaling. For the BMP signaling, it seems like it's the main signaling for the dental pulp differentiation. But for the bone marrow, it employs several signaling such as Wnt, TGF-beta, and BMP also. Anyway, you can find in this experiment result that canine dental pulp employ maybe mainly one of the signaling called BMP, but why they show the superior than bone marrow? Because the support of the evidence in mineralization at day 14, we found the canine dental pulp show the more mineralization area than canine bone marrow. And then when we found the Covary analysis result, we found a strongly expression of the protein in the group of ECM exaceramic expression in canine dental pulp. So in my understanding, the canine dental pulp is maybe play not much of the signaling of the osteogenic, but they play strong with this, very strong with this. So together with the maybe ECM collaboration, something like that. So you can show the result of the canine dental pulp superior than bone marrow. Mm-hmm.
Okay, thank you, Dr. Sirirat. Dr. Chenpop, you will add more information?
I think it's a very good answer right now.
Okay. Let me check. Is there any other question or not? I think.
Dr. Suryo, I think we have one question about the culture meat from some participants.
Oh, really?
At 9:36 a.m. and in the chat.
Oh, right.
Oh, okay. Yes. We have a question from Lanang, if I'm not, from Lanang. He's a student in our university. The first question is, I would like to ask how far the research and development stem cell for meat cell culture in Thailand. As we know, this study is prospective, especially to solve the environmental issue caused by farm pollution. And the second one, what is the most challenging to conduct meat cell culture research and development actually?
To answer this, let's say that it's different direction because we are making, or when we deal with their culture meat, we're making the food. So everything have to think differently. So first we have to set up the protocol to make a food. And I think the thing is that we try to mimic the environment of cell growth from natural muscle tissue into the lab setting. It's just that. And the way is we have to reduce the cost to make it eatable. And final, we have to produce it in the food grade facilities not in lab facility. Yeah. Big thing, just the budget. You need to find budget. Just that.
Okay. Thank you, Ajahn, for your explanation. I just remember because last 2 weeks or last week, our student contact me and asking about the lab-grown meat, but I don't have any idea because I don't have any experience about it. But I just remember, oh yeah. When I was there, you just start to develop the lab-grown meat with the company. Okay, maybe Lanang is one of the committee from the Indonesian IVASA, but I'm not sure. Okay, let me check the question again. I think we don't have any more question. Maybe for the last question for me to Ajahn Sirirat about the best practice, because you just introduced to all of us about the system biology, especially in proteomic approach, right? And to get the best result, to get the good result, the sample preparation is quite important, right?
Yes.
Can you share to us about the sample preparation, which step that is critical, and how did you handle in its critical step? Can you share to me, Ajahn Sirirat?
Okay. If the technique of the clinical of the sample preparation, the technique that I think it is the most carefully is about the cells preparation, because it is the generate the sample. So we have to calculate that how many cells that gonna get enough of the protein, because the protein we're gonna process to be a peptide and go to do the LCMS. So at the first time I failed about to prepare the cells because it's not enough the protein concentration. And you have to go back to the first step again for do the cell culture and then separate the protein homogeneous with the sonication, something like that. So the careful step is that you have to calculate first and make sure that you prepare the protein enough to do the mass spectrometry. And this is the general technique. And one more thing that I want you to keep in mind is it is the simple that protect your sample, but someone don't think before, is it your keratin on your skin? Is the keratin. So some lab don't strict to wear a glove or wear a cap. So it's gonna, if you imagine that your a little skin, if it like a add in your tube, it's gonna bigger than your protein. So when you analyze until in the end, you're gonna find that it is your protein from your skin. Okay, so please related. Careful. Yeah, please. I'm very careful in this step. Always wear glove, wear mask, also wear glove and wear the cap, because I want to protect my sample. And every time that I manage, I bring it to collect, to detect the peptide. Every time I'm gonna wear the glove and wear the scrub suit and wear the lab coat. Yes, this is very careful.
So it is the important things that we have to consider if we work with the proteomic, right? We have to avoid the contamination from our own protein, right?
Yes.
Yeah. So we don't have got the biased data. Okay, I think that's all for today's sessions. We don't have any more questions, but before I end this session, on behalf of the Universitas Airlangga, I would like to hand over the certificate of appreciation to you all. For the first certificate is awarded to Assistant Professor Dr. Chenpop Sawangmake, DVM, M.Sc, Ph.D. Thank you, Ajahn, for joining us today. And for the second certificate will be awarded to Dr. Sirirat Nantavisai, DVM, PhD. ขอบคุณค่ะพี่น้อง We are very happy to have you two in today's session. Okay. Before I end all of this today's sessions, let me try to recapitulate the message from our two distinguished speakers. Until now, we still face a big homework in terms of stem cell biology explorations, because a big mystery on stem cell biology is still remained. By understanding the basic concept of stem cell biology, will help us to provide the strong scientific evidence and also the proof concept, especially if we want to translate our finding into clinical applications. And from Dr. Sirirat, we learn a lot about one of powerful platform called as a system biology. In system biology, actually they have a lot of platforms such as a transcriptomic, proteomic, metabolomic, and another omic that can be used to help us to open the big mystery in stem cell biology. I think that's all that we can learn from these two speakers in today's sessions. Thank you for Dr. Chenpop, thank you for Dr. Sirirat, also for everyone for attending today's sessions. Thank you for joining us today and have a great rest of your day. And now I would like to hand over back my duty to the MC. Thank you, good afternoon, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Suryo, and all speakers, our amazing speakers, Dr. Chenpop and Dr. Sirirat, who have given us such a new light and fresh knowledge. I'm sure everyone in here feels glad with the presentation. And also, I'd like to greet the head of Veterinary Science Department from Universitas Airlangga, Professor Dr. Wijiati, Master of Science, DVM, Pafet K, and also the embryo lecturer from the Faculty of Veterinary Medicine, IPB, who also joined in this international webinar, Prof. Ari Budiono and Dr. Kustiantoro. Thank you so much for joining with us. And also all the participants who are in here in this Zoom session. But now we have come to an end of our session for today. But don't worry, this is a 2-day international webinar. So we still have tomorrow's session with the same topic, with the same theme, but with other competent speakers. So don't worry, don't miss it, and stay tuned. And before I end the discussion today, I would like to leave you with a quote from Richard Stallman, who once said, "Sharing knowledge is the most fundamental act of friendship, because it is a way you can give someone without losing something." Let us all be guided by all the things we have learned and heard throughout this international webinar. Let us keep on sharing our knowledge to one another. Thank you so much once again for our dear speakers and participants for participating today, and also Mr. Kusuryo as our moderator today. And I'm sorry if I made a mistake. Forgive me for my shortcomings. See you tomorrow. Stay safe and stay healthy. Wassalamualaikum warahmatullahi wabarakatuh. God bless you.
Salam satu. Thank you, Dr. Chenpop, Dr. Sirirat, Dr. Suryo.
Thank you very much, everyone.
Mrs. Debbie.
Ya, terima kasih banyak.
Take care.
Thank you.
Thank you, Napinong. Thank you, Najan.
Bye bye. Bye bye. See you.
Bye.
Bye bye, everyone. Bye bye. See you.
Bye bye.